Let's start from the beginning. In the digestive system, we know that we have three pairs of salivary glands, and those three pairs are the parietal gland, sublingual gland, and submandibular gland. They together produce saliva. Now, saliva contains amylase, and what does the amylase do? So the amylase basically over here breaks down the carbohydrates to oligosaccharides and disaccharides. So basically, if you look at a long chain of sugar, it breaks down into smaller one. So assume this is a long chain of sugar. After the amylase broke down this bond between them, as a result it produces disaccharides. So very long chain of sugar. Suppose there is a long chain of sugar that contains like number of sugars over here, and all the sugars are connected with each other. So the amylase does what? Break down this bond, break down this bond, break down this bond. As a result, it will produce oligosaccharides and disaccharides. So the sugar mixture of the carbohydrates is going to be in the stomach. From there, the food is going to go to the stomach. We know in the stomach, we discussed today, there are different types of cells we have. This is the gastric pit that you see over here. So within the gastric pit, we have the parietal cell, chief cells, and G cells. We know each one of them are producing different cells. Parietal cells produce hydrochloric acid, chief cells produce pepsinogens, and G cells produce gastrin. In addition to hydrochloric acid, so you see this one is parietal cell. Parietal cell, in addition to hydrochloric acid over here, it also produces a protein that's called intrinsic factor. This is important in absorption of the vitamin B12. So basically, if you look over here, the intrinsic factor, what does the intrinsic factor do? Intrinsic factors enables vitamin B12 absorption. And HCl, what does the HCl do? HCl converts pepsinogen to pepsin, as we discussed today. It denatures protein, it kills microorganisms, breaks connective tissue. We know that the chief cells over here produce pepsinogen, and pepsinogen is the inactive form of pepsin, and that's the proenzyme. So it will be converted to pepsin in the presence of acid. So once pepsinogen is converted to pepsin over here, so pepsinogen is converted to pepsin by the acid, which is produced by the gastric lumen, and pepsin is digesting the protease and degrades the food protein into peptides. So the basic function of the pepsin is to break down peptide bond and break down the proteins. Let's look at the G cells. G cells, we said, produce a gastrin. Now remember that gastrin over here is not it's a hormone, it's not an enzyme. So gastrin is a hormone that's produced by the G cell. And what is the function of the gastrin? Gastrin stimulates secretion of the parietal cell and also the chief cell. So basically, it's the gastrin that actually stimulates secretion of the HCl and pepsinogen by the chief cells and parietal cells. So why does the absorption doesn't take place in the stomach? We said the absorption takes place in the intestine. Number one, that the parietal cells are covered by a blanket of alkaline mucus, and they are not directly exposed to the chyme. So number one is that they're not exposed to the chyme. Number two, the epithelial cells lack this specialized transport mechanism. They don't have this specialized transport mechanism to transport the food into the circulation. The third one is the gastric lining is relatively impermeable to water, so the water cannot pass through this one. And digestion has not been completed. Remember, we said digestion of fat and carbohydrates continue in the small intestine. Because digestion is not complete, it's not able to absorb it. However, you should remember that some drugs such as aspirin could be absorbed by the stomach. Okay, after this one, once all this process occur in the stomach, the food is going to get where? To the first part of the small intestine, that's the duodenum. So the arrival of lipids, carbohydrates in the duodenum, this will stimulate what? This will initiate or stimulate production of two hormones, and these two hormones are the CCK and GIP. So the CCK and GIP are produced when the lipids and carbohydrates arrive in duodenum. Also, when the food arrives in duodenum, a pH will drop because we said the content of the stomach is acidic. So a drop in the pH below 4.7, this will stimulate secretion of another hormone that's called secretin. So basically, arrival of food in the duodenum will stimulate secretion of CCK, GIP, and secretin. So secretin levels in the blood will increase. It will initiate number of things. One of the things it will increase viscosity of the intestinal polypeptides. It will increase polypeptide hormone secretion by the cell throughout the intestine, and so forth. So we're going to look at number of those in a second. So those are the effects. So what is going to happen? The food gets into the duodenum. 1, 2, 3 things are secreted. Now let's look at each of those 3 things that are produced. First, gastrointestinal peptide. Gastric inhibitory peptide, GIP, inhibits gastric secretion. Remember, we don't want the stomach to keep producing the acids and everything. So GIP will tell the stomach. So remember, the food is now over here. So right over here you have the stomach. So this GIP will go from here and it will tell the stomach, stop producing acid. We don't need you to produce any more acid. And also it will stimulate release of insulin. Remember, when you eat food, the concentration of glucose in the blood increases, and that stimulates release of insulin. Okay, let's look at the second one. Secretin inhibits parietal cells and chief cells activity in the stomach. So also secretin does what? Secretin tells the stomach, goes from here and tells the stomach, don't produce any more hydrochloric acid, don't produce any more pepsinogens, and so forth. So it inhibits activity in the stomach. Also it stimulates production of buffer. That buffer is called actually bicarbonate. So is over here you have the pancreas. Also secretin goes to the pancreas and tell the pancreas, produce some buffer, because the pH in the duodenum is acidic, and that acidic pH is not good. So produce some buffer to neutralize that pH. Now let's look at the next one. What does the next one do? So the next hormone is over here, CCK, also produced by the intestine. As soon as the lipids and carbohydrates get into the intestine, this is produced. What is the function of this one? This also inhibits gastric secretion. So it will also go to the stomach over here, and it will tell the stomach what? Don't produce any more acid, don't produce any more pepsinogen. And this hormone also will go over here. You have the gallbladder, and over here you have the liver. So the liver produces bile, it goes to the gallbladder, stored in the gallbladder. So this hormone will go from the intestine and tell the gallbladder to contract, so that the content of the gallbladder bile is delivered to the intestine. So it stimulates gallbladder, and the bile is as a result ejected into the duodenum. Now the brush border of the intestine also produces other enzymes, such as produces maltase over here, and produces sucrase, the second enzyme, and lactase, the third enzyme. So it produces maltase, sucrase, and lactase. Sucrase basically maltase breaks down the maltose, sucrase breaks down the sucrose, and lactose breaks down the lactose. Those are all sugars, carbohydrates. So different carbohydrates are broken down. Remember, what is the goal? To break down everything into a single sugar unit. So we started with a long chain of carbohydrates. The digestion begins in the oral cavity, it comes to the stomach, and then the intestine. So it continues with the breakdown of carbohydrates. So we said that the secretion of the bile duct, from the gallbladder and from the pancreas is going to go. So this is the common bile duct. So the secretion from the gallbladder will come through this duct, and from the pancreas it will go through this, and everything is going to be mixed in the intestine. So let me start back. So the pancreas produces the pancreatic juice, and the pancreatic juice comes through those tiny ducts over here, the accessory pancreatic ducts, delivered all through this duct, and it goes to the duodenum. And if you look at those ducts, this duct, the green one that you see over here, this is coming from the gallbladder. That's the common bile duct. And those tiny ducts are from the pancreas. Now remember, all the ducts are joining together over here before they're emptied into duodenum. So what does the pancreas produce? Pancreas producing a lot of juices. Some of the enzymes that is important that we're going to mention over here is we said pancreatic amylase that digests what? Pancreatic amylase digests the carbohydrates. Peptidase digest the peptides. Chymotrypsins digest the peptides. Trypsin digests the peptides. Lipase digest the fat and other enzymes. So it produces a collection of enzymes. As we mentioned earlier, the secretion of digestive enzyme, the secretion of the pancreatic juices are controlled by CCK and secretin over here. So you see those two hormones, they're actually controlling the secretion of the pancreatic juice, and the pancreatic juice works in the small intestine to digest most of those foods. So amylase is an enzyme that breaks down the certain starches. Lipase breaks down certain complex lipids, releasing products such as fatty acids. Protease, e.g. trypsin, breaks down certain types of proteins. Let's look at the gallbladder. We said the secretion of the gallbladder is stimulated by CCK, a hormone produced by the small intestine. So when the food gets to the small intestine, CCK is produced. CCK goes to the gallbladder and stimulate production of bile. What is the gallbladder? Most dietary lipids are not water soluble, so mechanical processing in the stomach creates large droplets containing a variety of lipids. Pancreatic lipase, on the other hand, is not a lipid soluble. As we mentioned today, it's an enzyme which is water soluble, so it would not be able to penetrate inside the large droplet. So what is the function of gallbladder? Gallbladder stores bile and it modifies biles produced by the liver. And bile salts, what is the function of the bile? Bile salts produced over here, you see it, the bile salt breaks the fat droplets apart in a process called emulsification. And what is emulsification? Emulsification basically does what? It increases the surface area of the fat droplets, and therefore it makes it accessible to the enzyme lipase, so that the enzyme could attack it and break it down. Hope this helps you, and have a nice day.